Hello students, welcome back. Today we are going to discuss some more concepts from principles of inheritance and variation. Today's concept is chromosomal theory of inheritance. Mandela published his work on inheritance of characters in the year 1865. For several reasons, it is remained unrecognized till 1900. So now let's see that what are the reasons it leads to remain unrecognized till 1900. The reasons unrecognition of Mandel research till 1900. The first reason is communication was not easy in those days and his work could not be widely published. Second reason, his concept of factors, nowadays it is called genes, as stable and discrete units that controlled the expression of traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries. Third reason, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Fourth reason, though Mendel's work suggested that factors were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. So just now we have studied about the reasons why the Mendel's research is not recognized till 1900. So now let's in 1900, three scientists, De Vries, Correns and Testmark, independently rediscovered Mendel's results on the inheritance of characters. Why they are rediscovered in those days, that is in 1900? Because by this time, due to the advancement in microscope, what takes place? Scientists were able to observe cell division. This leads to discovery of structures in the nucleus that appeared to double and divide just before each cell division. These were called chromosomes. That means the structures which are present in nucleus that are appeared to double before the each cell division. That structures which are doubled before each cell division, that structures are called as chromosomes, which are located in the nucleus. By 1902, the chromosome movement during meiosis had been worked out. So due to that reasons, that means due to advancement in the cytology, cell biology, then these three scientists, De Vries, Correns and Testmark, they start to rediscovering the Mendel's research. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. So already in previous slide we discussed by 1900 the development in the cytology that is used by this Walter Sutton and Theodore Boveri. So they noted the behavior of chromosome was parallel to the behavior of genes. Both are parallel. And used the chromosome movement to explain the Mendel's laws. So let's recall the behavior of chromosomes during mitosis and during meiosis. So once recollect the concepts of that cell divisions, so which we are studied in first PUC, that is 10th chapter. Chromosomes as well as genes occurs in pairs. Two alleles of a gene pair are located on homologous sites on homologous chromosomes. So let's recollect about cell division. We are all very well known that there are two types of cell divisions, mitosis and meiosis. Let's see about mitosis. So mitosis takes place in two stages. Don't confuse two stages. That is karyokinesis, and the second stage is cytokinesis. So now let's see that what is meant by this karyokinesis. Karyokinesis is nothing but a nuclear division. So this nuclear division or a karyokinesis occurs in four phases, that is prophase, metaphase, anaphase, and fourth one is telophase. Are the four phases. And cytokinesis, after completion of this nuclear division, that means at the end of this telophase, the cytokinesis is initiated, then cell divides into two. Now let's start the brief discussion of this prophase, metaphase, anaphase and telophase. So now prophase. So during this prophase, what are the major things we need to observe? That is, one is during prophase we will observe the structure of chromosomes. We can observe clearly. So that means chromosome having two arm-like structure, chromatids, and they are joined at the center called centromeres. And the next one is that disappearance of this nuclear membrane, disappearance of nuclear membrane and nucleolus takes place during prophase. Now let's come to the metaphase. So during this metaphase, all the chromosomes, centromeres of chromosomes are arranged at the center called as equatorial plate. Now let's see the third phase, anaphase. So during this anaphase, the chromosomes are pulls to the opposite poles. Opposite poles. So you may have doubt that how they are pulled. Now let's come to prophase and anaphase. So there is a special structures in animal cell that is called as centrioles or the organelles. These centrioles produces these spindle fibers. Spindle fibers. So these spindle fiber formation is initiated in prophase, and these spindle fibers are attached to the centromeres during metaphase. Now these spindle fibers pulls these chromosomes into the opposite poles. Then come to the telophase. So during this telophase, already the chromosomes are equally distributed to the opposite poles. Then whatever they disappear during prophase, they are reappears during telophase. That means reappearance of nuclear membrane and reappearance of nucleolus takes place during telophase. Now, after this reappearance of this nuclear membrane and nucleolus, and then the cell undergo cytokinesis. Cytokinesis. As a result, the cell divides into two daughter cells. So now, here during mitosis, we need to remember a key point is that before starting cell division, the chromosome number is doubles during mitosis. Before starting cell division, chromosome number is doubled. So this we need to remember. It is happen during mitosis. So let's come to the meiosis. So this meiosis is occurs two parts. One is meiosis 1 and meiosis 2. And in meiosis 1 as well as in meiosis 2, it is occurs in two stages, that is karyokinesis as well as cytokinesis. It is common, and in during meiosis 2 is also occurs through this karyokinesis as well as cytokinesis. So during cell division, this karyokinesis and cytokinesis is common. Nuclear division and cytoplasm division, it is common. So during meiosis 1, the karyokinesis is considered as karyokinesis 1, cytokinesis 1. During meiosis, karyokinesis 2 and cytokinesis 2. So now let's count up the karyokinesis 1. Karyokinesis 1 is occurs in four phases, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. And after completion of this telophase 1, then it leads to cytokinesis 1. And in this prophase 1, again subdivided into 5 types. that is zygotin, leptotin, zygotin, zygotin, leptotin, 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 zygotin. Here it is exchange leptotin, zygotin, packaging, packaging, diplotin, diplotin, and finally it is diakinesis. Diakinesis. So during meiosis 2, the chromosome not doubled. So now let's come to that. What will happen during? So during prophase 1 of meiosis 1, prophase 1, during prophase 1, prophase 1, already we are given 5 subphases we are observed during prophase 1. It consists 5 subphases. It is one important point in this slide. 5 subphases. During these 5 subphases, crossing over of chromosomes takes place. Crossing over of chromosomes takes place. Remember that this crossing over again we will discuss in the next part. So crossing over takes place during prophase 1. And like mitosis, here also chromosome structure is identified as well as disappearance of nuclear membrane as well as nucleolus is also takes place. So metaphase 1, anaphase 1 and telophase 1, what we are discussed in mitosis is same. So centromeres are arranged at the center equatorial plane. Pulling of chromosomes, it is happened during anaphase, and reappearance of nuclear membrane and all, it is happened during telophase 1, and finally cell divides into 2 daughter cells. So after completion of cytokinesis 1, so forms 2 cells, daughter cells are formed. So each daughter cell gets the exact of the number of chromosomes. That's why they are haploid in condition. Chromosomes are reduced to half. So that's why this meiosis 1 division, is called as meiosis 1 division, is also called as reductional division. Reductional division. So the chromosome number is reduced to half. Now, the 2 daughter cells which are formed during meiosis 1 these 2 cells undergo meiosis 2 cells. So now this meiosis 2 is called as equational division. Equational division is nothing but mitosis. It is similar to the mitosis. It is similar to mitosis division. So in prophase 1, here we are said that it is having 5 subdivisions or phases. But whereas in prophase 2 there is no subdivisions. So here also same, chromosome structure identified, disappear of nuclear membrane and all. And now 2 cells are involved, and metaphase arranged in equatorial plate, anaphase pulling the chromosomes, telophase, so reappearance of disappeared things in the prophase, and cytokinesis. Now 2 cells are involved. Each cell produces 2 cells. Each cell produces 2. So that means 2 into 2. Total 4 cells are produced at the end of meiosis. Meiosis 1 and meiosis 2. After completion of these two, total 4 daughter cells are formed and all the 4 daughter cells are in haploid condition. This concept we discussed in the first PU. This is a brief. So in meiosis we will observe that two type divisions. One is reductional and equational. That reductional division meiosis 1, equational one is the meiosis 2. So during meiosis 1 is a very crucial. In that meiosis 1, the crossing over of chromosomes takes place. When this crossing over takes place, takes place, the genetic recombination takes place, exchanging of genetic material takes place during the crossing over. That leads to the appearance of new characters. That's why in this inheritance of chromosomal theory of inheritance, the cell division is important. And already we said that the discovery of Mandela's is unpublished till 1900. In 1900, the cell biology developed well due to development of microscope. As a result, all these changes are studied. Based on these studies, they applied this cell biology on that genetics. And this Theodore Boveri and Sutton given this theory, that is chromosomal theory of inheritance. During anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of 4 different colors in the possibility 1 and possibility 2. In possibility 1, orange and green chromosomes are segregated together. Remember that orange and green, that means that to identifying the difference, we are applied some color to that. So meiosis 1. So you are all very well studied. During anaphase, the chromosome pulling takes place. So chromosomes are pulled to the opposite poles. As a result, half of the number of chromosomes are moves to the opposite poles and later it leads to the telophase. So now in possibility 1, orange and green chromosomes are segregated together. So now here in this possibility 1, in this diagram, the orange and green chromosomes are segregated together, and in another cell yellow and red chromosomes are segregated together. So let's see the next. So now in meiosis 2 it is same, again it is pull it. Again it will pull it. So that means in anaphase 1, one cell will get this orange and green chromosome and another cell will get this yellow and red chromosomes. And in anaphase 2, during meiosis 2, again these two cells are undergo cell division and each cell will get, two cells will get orange and green chromosomes and two cells will get yellow and red chromosomes. See, this is, it is separated like this. So then see the diagram. So now in germ cells or in gametes it will get only one set, that is orange and green color chromosomes only present in each cell. So here it is paired and that is separated during gamete formation. Let's see the possibility 2. The orange chromosome is segregating with the red chromosome. There is another possibility. So orange chromosome is segregated with the red chromosome and yellow chromosome is segregated with green chromosome. Segregate means separated with green chromosome. So in meiosis to anaphase, each cell will get orange and red chromosome pair and another cell will get yellow and green colored chromosome. And in gametes, this is the combination. In first possibility, orange and green. And in second possibility, yellow and green, orange and red combination is the possibility of the cell division. Sutton and Boveri argued that a pairing and separation of a pair of chromosomes would lead to the segregation of pair of factors they carried. So whatever the pair of chromosomes separated a pair of chromosomes it leads to the separation of pair of factors they carried. Pair of factors nowadays they are called as genes. A pair of factors they are carried are also separated along with the separation of chromosomes. Sutton and Bowley independently proposed the chromosomal theory of inheritance in 1902. Chromosomal theory of inheritance was modified and expanded by Morgan, Stuyvesant and Bridges. These three scientists or modified this chromosomal theory of inheritance proposed by Sutton and Boveri and modified by Morgan, Stuyvesant and Bridges. Let's see the silent features of chromosomal theory of inheritance. Genes or genetic information is carried by chromosomes as per this chromosomal theory of inheritance. Genes or genetic information is carried by the chromosomes. Genes are located on specific location on the chromosomes called locus. So what is meant by a locus? Genes are located on a specific location on the chromosome called as locus. Chromosomes are located in the nuclei, thus indicating that nuclei play an important role in hereditary. That means characters transferring from one generation to another generation. That the important role played by a cell part called as nuclei, because nuclei consists chromosomes. A typical body cell is diploid, which implies that it contains two sets of genes. So, if any cell is in diploid condition, that indicates that the diploid cell consists two sets of genes. The gametes are haploid, containing one set of genes. Thus, during gamete formation, the number of chromosomes is reduced to half. The gametes fuse during fertilization and contribute equally to the heredity of the offsprings. The chromosomes follow Mendel's law of segregation and independent assortment during meiosis and recombine at the time of fertilization in the zygote. Thank you, students. We will discuss a last concepts from this chapter in the next part.